Hello, my name is Akis Jackson. I am a junior at Oglethorpe University. This is my bioseminar long talk. My presentation is on the maternal manipulation hypothesis, reptilian viviparity versus oviparity. What brought me to this subject is that I've always had a fascination with snakes or reptiles in general. And so when looking for something to present on, the name maternal manipulation stuck out to me. And so firstly, we have the outline of the presentation. I just gave you a small background, and I'm going to give you a few keywords that will pop up repeatedly throughout the presentation. I will address each study individually, and in that I will give the research question, hypothesis, the approach, and the results. And then near the end, I will compare the experiments a small bit, and then give you a take home. All right, so key terms and ideas. The first paper / experiment deals with the short-tailed pit viper, Lloydius brevicaudus. The second paper, second experiment, deals with the eastern three-line skink, Bassiana duperii. All right. Next, the maternal manipulation hypothesis, what both these papers are based off of. This hypothesis hypothesizes that mothers who are pregnant or carrying eggs or have laid their eggs will manipulate their body temperature or the temperature around them to give their offspring the best phenotype, the best expression of traits. Viviparity refers to any live birth that occurs from an organism. Oviparity refers to giving birth to eggs. Rabbit means pregnant or carrying eggs. Neonates refers to babies or offspring. New offspring, babies, yes. Oviposition refers to the act of giving birth to eggs. Phenotype is the expression of genes, what an organism outwardly looks like and performs. And fitness. Fitness is used in both these tests as a marker of how well the phenotypes have been expressed. And fitness can mean many things, but in both of these experiments, it was given to a few traits such as running speed, swimming speed, snout vent length, tail length, head width, things of that nature. All right. So firstly, the evolution of reptilian viviparity. Our first research question is, does the maternal manipulation hypothesis apply to temperate reptiles? This is the question because this study has already been conducted on colder climate reptiles and tropical climate reptiles. So now it's time for the temperate. The temperate is in the green area you can see on the map. This snake in particular, the short-tailed pit viper, is native to Manchurian China, China and the Korean Peninsula. The hypothesis is that thermoregulation during development improves offspring fitness. So they're again trying to prove the maternal manipulation hypothesis, but with temperate reptiles. And the question mainly is, even though we've tried temperate or we've tried tropical and polar, why this animal? And the answer is because the temperate climate or the temperate area of the Earth can vary in temperature throughout the year, very much so. And so it is both a good midpoint, but also a good sense of how much variation could go on while offspring is incubating in eggs or in the ground wherever the nest may be, or inside their mother. All right, for our first experiment. So firstly, they had a total of 80 snakes. And out of these 80 snakes, only 14 were not pregnant. The rest of them were chosen out of nature to be pregnant, or they were ensured to be pregnant and then chosen. All right, for the first three groups, they were very similar. The goal of these groups were to keep the snake's mean body temperature as close as possible to these temperatures. And so the first one was at 22 degrees, the first group of 12. The next group of 12 was at 27 degrees, and the last group of 12 was at 32 degrees. Next, there was the temperature regulator room. This room, the snakes had gradients in each of their own little cells, in which the ambient room temperature was 20 degrees Celsius, and where they were in their little cells had an area of 20 degrees Celsius, but also on the other side of that was an area of around 55 degrees Celsius, which gave the reptiles ample opportunity to move, adjust, reheat, cool down, as they needed and wanted. This was also group 12. Next, we had the field treatment. These were a group of reptiles who were picked from the same area around the same time, early September. Not around the same time, excuse me. They were picked in early September. And they all gave birth within a week of being introduced to the lab. And they did this so they would see how how nature, thermoregulating in nature, compares to the rest of these, not so much as to the lab. And finally, there was a separate preference group. This group was a group of 22 total. There was 7 females who were not pregnant, eight, excuse me, 8 pregnant females, and 7 males. And again, this was just to see how the snakes preferred the temperature preference. They were given the same or the same gradient as the temperature regulated room. And I know you may be asking, the range kind of makes sense, the field treatment kind of makes sense, but why these selected temperatures, this group of 3, why are these temperatures? And so I'll show you. For figure 1, we have the mean temperature variation in the viper's habitats in the areas where the field treatment and the rest of the vipers were collected from. And as you can see, this graph runs from late June to mid-September. And the mean line is the solid black line that varies very much in the middle between the maximum and minimum lines. And as you can see, the lowest point in the mean line is around 22 degrees and the highest point is around 32 degrees. That is why these two are our lowest and highest attempts at getting the viper body temperatures. And 27 is in the middle because -5 +5. And so the horizontal lines that run across show where the average body temperature of the snakes never got below, the average body temperature. And so the top horizontal line, 28.7 degrees, shows that the maximum viper body temperature, excuse me, on average, never got above 28.7, never got below, on the bottom line, 24.9. All right, table 1, the statistics of female reproductive traits. So on the graph, there has been some data cut out for the effort of taking up much space that we don't need. We have each of the 5 treatments: field, temperature controlled, 22 degree, 27 degree, 32 degree. The 32 degree room had the highest snout vent length, the highest litter size, but unfortunately, it also had the highest group of deformed offspring and stillborns. With these, usually temperature controlled room and the 27 degree room tend to do okay with these measurements, excuse me. And the proportion of deformed offspring that has the absolute lowest is the 27 degree room, while the 22 degree room is the second highest for the proportion of deformed offspring. All right, for figure 2, here we have the range and average amount of days from the 1st of August. And so this is showing us, again, the average amount of days from the 1st of August that each control group on average took to be born, or not control group, excuse me, each group. And so as you can see, 22 degrees took the longest at an average of 94 days, while 32 took the shortest on an average of 18.22 days. Excuse me. Here we have 27 degree, the field treatment, and the temperature controlled room treatment. All of these are somewhere near the middle, but the temperature controlled room seems to have the largest range of the average of dates, while the field treatment seems to have the very most shortest. And so, this may bring the question, is there a correlation between the temperature and the time it takes to incubate and be born? And the answer is yes, there seems to be a correlation. However, that also brings up, is it good or bad taking too long or not long enough for offspring's development? All right, next we have one of the traits of fitness I mentioned earlier, snout vent length. This was a literal measurement of the vents on the snouts of this offspring snakes. And so all of them correlate with snout vent length. These were originally one picture, but they've just simply been split so they can all be shown at the same time. Snout vent length positively correlates with litter size, litter mass, and neonate mass or offspring mass. It seems to correlate the most with litter mass, as that is the seemingly tightest packed group of data points, and the least amount with neonate mass, but those are the most spread out. Very close to litter mass, litter size, excuse me, I would say. So yes. All right, next we have figure 4 and figure 5. Figure 4 shows us how thermal treatments affected the offspring's swimming speed and stamina. And so after the offspring were born, they had to be tested, of course. And so what they did was they tested the mass and the snout ventilating, as mentioned, of the snakes, but they also put them in a small container with of a certain length with water, and they nudge them with the paintbrush to see how fast they swim and how long they could continuously swim. And thus they have swimming speed and stamina the amount of time they swim. And it seems the 20 to 7 degree treatment did the best in both cases, both in swimming speed and stamina. With figure 5, this shows the growth of the newborns, the neonates, 90 days after birth. And on average, all of the groups grew in mass except the 32-degree group, which again, that might be a sign that this high temperature and fast growth may not be the best for actual development and fitness of offspring. All right, so for the results, we confirmed the hypothesis. Um, the thermal regulation of a mother's body can be beneficial to offspring. The temperature extremes of 22 degrees and 32 degrees are both bad for offspring. They're also bad for the mother, as this can lead to the mother trying too hard, exerting too much energy, even dying, as there were a number of snakes who did pass away in this experiment. These temperature extremes can also cause deformation, stillborns, and things of that nature. And so by extension, we could actually test and ask, what other animal species other than reptiles can this be tested on? And if, push comes to shove, temperatures over the world start ranging wildly, either increasing or decreasing. Could this lead to a loss of species if too many offspring are born deformed or stillborn, or some kind of mutation or bad expression of the phenotype that doesn't allow them to mate to live? What would happen? And next, it is time for the second paper. And so, the maternal manipulation of offspring phenotypes via nest site selection. So here we have the eastern three-line skink, Bassiana perii, excuse me. And the research question is, what effect does thermal variation have on offspring that rely on incubation sites? And so we know that thermal regulation has an effect on can have an effect on the offspring, but what about thermal variation? What about the actual change, what effects do the actual change in temperature have on the offspring? And so the hypothesis was mothers may be able to affect the phenotype of their offspring by choosing the best temperature for incubation. Again, we're trying to prove the maternal manipulation hypothesis, but this time with oviparous, with egg birthing animals. All right, for the methods. So firstly, the researchers found areas of nesting and areas of what seemed like good nesting sites, like very similar sites, and they put thermal detection, temperature detecting technology there to track the fluctuation in temperature in these natural sites. They also gathered many gravid, pregnant reptiles and brought them to a lab for testing. This experiment took place over multiple years. And so for the first year, they conducted They conducted experiments to find the mean incubation temperatures for a clutch or a group of eggs at 22 degrees and 30 degrees. Both of these are in Celsius. And they were kept at these constant temperatures just to see the difference. For the second year, they measured the variations in incubation temperature while keeping the mean temperature constant. Basically, how does the change in temperature affect the offspring? And so for this, there were three areas. There's a group held at constant 23 degrees, and 23 degrees was the baseline where the variations were based around. For the second group, they were kept on a daily 10-step cycle of thermal variation with the 7.5 degree range. This went to 19.25 degrees Celsius to 26.75 degrees Celsius. And for the final group, they were kept on a 10-day step cycle as well, or daily 10-step cycle for thermal variation as well. They were kept on a range of 19.5 as a whole. At the lowest, 13.25 degrees Celsius. At the highest, 32.75 degrees Celsius. There's even more methodologies to this. And they also had a separate group of eggs, which they incubated at different constant temperatures, just to see if there's a relationship between, even more so, how long incubation periods take, and if they're related at all to incubation temperature. This paper did occur before the short-tailed pit viper experimentation paper. They also did was put siblings in different temperature treatments. And this, of course, was to see how similar genotypes were affected by different temperature conditions. And the experimental results from both of these years were analyzed separately and then compared. This is because the scientists wanted to make sure that any small thing they might not have noticed doesn't affect the results, whether it be something different within the lab, something different within nature, something different within the way they are handling the eggs. They wanted all of those things, all of those variables they might not be able to account for, to stay out of the experiments. All right, for measurements. Incubators were checked daily and the hatchlings were removed, weighed, placed in separate containers, and given food, water, and heat as they needed and wanted. And at 7 days of age, each hatchling was reweighed and tested for their running speed. Again, they were nudged with a paintbrush along a contained pathway. The time was measured. The test room was at 25 degrees Celsius, and they were each in all giving an hour to acclimate to the room. Right? Measurements continued. At 10 days old, they also had their activity patterns measured. Ambient temperature in the room, in that room, was maintained at 25 degrees, and videos were taken to score the basking frequency every 10 minutes. This was meant to keep a track of how often each group tended to need to go bask in warmer light or warmer temperatures to raise their body temperature as needed. All right, for figure 1, we have the mean incubation temperatures of 2 randomly selected nests. And as you can see, the variation for the top nest tends to be on the decline very slightly, while the bottom nest seems to be all around steady, with only a very, very slight increase in the beginning, or overall increase, excuse me. And so the variation, as you can see from bottom to spike, day by day, can be at most around and over 30 degrees Celsius. They can be around and smaller than 10 degrees Celsius as well. And it's also important to notice that these measurements were taken over a 60-day period, not just a very short period. 60 days, around 2 months. Next, we have the incubation temperature correlations. And so we see that there's a positive correlation between incubation temperature and the development rate, as we saw in the last experiment. That was proved after a few decades after this one. Excuse me. Incubation temperature raised, or development rate raised with the incubation temperature. The faster the development rate, there was probably a higher temperature in these nests and areas. However, there seems to be a negative correlation between incubation temperature an incubation period, which again means the longer it took to incubate, the shorter amount of time, excuse me, the longer it took to incubate, the higher the temperature tended to be. The higher the developmental rate, the higher the temperature tended to be. All right. For table 1, we have the effects of 22 degrees and 30-degree treatment. All right, as you can notice, there are between the constant temperatures, excuse me, percentage of time basking, the masses, and the snout vent lengths were all very similar. However, for the 22-degree treatment, the incubation period was longer. There was a lower survival rate to hatching and a lower ratio of males to females. Also, all the way down here at total activity, at 1 month, the 30 degrees Celsius tended to have much more total activity than the 22 degrees Celsius treatment. For many of these, they are somewhat similar, but the variation, the uncertainty, as you see next to them, puts them within the same range usually. And now the effect of variation in temperature. And so here, the high variance treatment reduced incubation period. 23 degrees constant was around 53. So was the 23 degrees with a small variation of 3.75 either way. But the 23 degree with 9.75 variation either way decreased by almost 20% at around 41 days. There was also a difference in mass at hatching, with both of these being around 270 and this one being a little lower. There's not much of a difference when you realize that the variation of 3.75 is very similar, but there still is a marked difference. As well as in the tail length, there is a very heavy increase of the 23-degree group compared to the others. But with total activity and total time spent basking, the 23-degree group with the highest variation tended to have the lowest scores, much lower than others. All right, for our results, we confirmed, again, the maternal manipulation hypothesis. Temperature variation also does have an effect on the observed traits of the hatchlings. Developmental rates seem to be linearly related to incubation temperature. And when we compared siblings who went to different temperature treatments, hatchlings from the higher temperatures were longer, they ran slower, and they spent less time basking and less time being active. And so, time for comparing the experiments. Both of these confirmed the maternal manipulation hypothesis, but also almost inadvertently, if not, well, not inadvertently because they did test for it, both showed a relationship between temperature and incubation. And the higher the temperature, the shorter the incubation, and vice versa. However, they also show that with these variations in incubation come traits that are traits and phenotypes that are poorly expressed, that come high rates of stillborns or deformities, things of that nature. And so also, another question to identify with this is, if the maternal manipulation hypothesis is proven for both of these, why certain reptiles have chosen, or not chosen, but have evolved to give birth to eggs versus live birth. And so egg birth is would be a little more difficult. Excuse me, not difficult. Each option has their own benefits and downfalls. The egg option there is difficulty in finding a good site, and if it is a type of species of reptile that will stay with the eggs and monitor them while they incubate, they need to find a site that they can regulate the temperature of by adding and taking away debris, mass, whatever it be. maybe. Um, they also have to keep guard over them, things of that nature. However, there might not be as much energy spent um, with the eggs or with the embryos if it was the other way around, going inside of them. As humans often say, eating for two, after you lay your eggs and they are in their incubation site, after they're in the nest, um, it might not take as much effort to keep yourself energized, alive, things of that nature. With live birth, with with live birth, mothers are more able to regulate their body temperatures and thus affect the phenotype of their offspring. It is much easier to regulate an embryo inside of your body than a group of eggs in an environment. However, with repeated live birth comes an effect on the mother's reproductive organs. They tend to work worse, somewhat degrade in some cases. So yes, take home, the maternal manipulation hypothesis has been proven correct either way. You know, heat in certain reptiles, such as crocodilians and turtles, does have an effect on gender. But through both of these studies, we see that it also has an effect on phenotypes, how genes are expressed, these fitness traits, how well the animal moves, runs, um, what it needs to do to regulate its body temperature, more or less. And it has an effect on fitness. And again, fitness in this case was given to certain traits that they picked out: head length, tail length, snout vent length, running speed, and swimming speed, things of that nature. And when we can again ask the question about humans. Does the hypothesis apply to humans where thermal regulation of a human mother and her baby will that improve the baby's fitness in the long run? And in general, how do temperatures affect human embryos? While a human is being formed inside of their mother, is there any kind of correlation between where the baby is born in terms of heat, as in a very cold climate versus a really warm climate? Is the baby more fit in either one, or is it solely fit to be adaptive? Is it solely fit to be adapted to their climate? Thank you very much.